Welcome to my coronavirus classroom. My name is Janessa Jacobs, and this is blood vessel physiology. So we left off talking about the anatomy of blood vessels, and today we're going to talk about the way that blood flows through each of our different vessel types. So we have to think about the pressures acting in the system, the relationships of those pressures to blood flow, and what it means for all of our vessel types. So everything in life is guided by physics and chemistry, and blood is no exception. When we think about flow of any kind of fluid, we need to think about the pressure that it's under and then the resistance to that flow. So the blood is no different. So let's think about blood. Blood flow is the flow of blood through an organ, a vessel, a tissue, or the entire system at any given moment that we can measure. So blood flow is blood flowing through a tissue, vessel, organ, or the system at any given moment. And the flow of any kind of fluid can be calculated. I'm not really going to have you calculate equations, but we need to be aware of the relationship between this blood flow and the pressure in the system and the resistance in the system. So the flow of any fluid is guided kind of by a change in pressure or a difference in pressure, a pressure gradient, which is, this is delta P, and then divided by the resistance. So mathematically, what this means is that there's a direct relationship between the difference in pressure between one area and another and the blood flow to that area, and an inverse relationship between the resistance to an area and the blood flow to that area. So what that means is as the difference in pressure goes up, as we've got a difference from high pressure to low pressure, we'll have more blood flow. And as we have more resistance, as we go, let's say, on a longer pathway or something like that, then you have less blood flow. So what does this matter and why do we care? Because it's going to help us figure out what's going on in the system. So blood pressure specifically is the pressure that blood is exerting on our various vessels. When you have your blood pressure measured and they're taking your systolic over your diastolic blood pressure, what they're measuring is your arterial blood pressure. And we'll talk about that in a minute. But for blood pressure as a general idea, what is blood pressure? It's the pressure of blood on a vessel at any given moment. Now this is the pressure, a physical pressure of a fluid on a membrane. So that we call hydrostatic pressure. And this is an outward pressure in the arteries due to ventricular systole. So blood pressure is this pressure of blood on a vessel, and this is the hydrostatic pressure of blood on that vessel. Okay, resistance is just the opposition to flow. So what we're trying to have is blood flow, and we always want to be able to maintain blood pressure so that we can have blood flow. Blood wants to flow from high to low. And so these are all kinds of things that we have to put together to think about what's going on in the system. So your highest pressure due to ventricular systole is in the aorta. And so that pressure is going to be really high. The lowest pressure is when you is at the superior and inferior vena cava. Or the pressures acting on venous blood returning to the heart is different than the hydrostatic pressure due to ventricular systole. So we'll look at all that in a minute. But one of the things that we're going to see is that when your blood pressure drops, the body has neural and a lot of hormonal mechanisms that can be engaged right away to bring your blood pressure up. Because if your blood pressure drops, your blood flow drops. If your blood flow drops, you die. So it's really, really important to be able to keep blood pressure up. And there are really only a couple things that happen when your blood pressure is high for a long time. Because really, I mean, yeah, it's going to stress your cardiovascular system, but it's not deadly as quickly as losing blood flow. So that's really what we're going to focus on, is the relationship of blood pressure to blood flow. But we just need to be aware that as we encounter resistance, that's going to decrease blood flow in the system. Okay. So there's probably a figure in your book that shows something like our pressure differences throughout the system when you go from the aorta and our pressure is the highest to when you return to the superior vena cava and the pressure is the lowest. So have a look at that and relate that to everything I just told you, and it'll make some sense. Okay, so what are the pressures on the blood in our different types of vessels? Our arterial pressure is the hydrostatic pressure due to ventricular systole when our ventricles are in systole, and that it is the pressure, still hydrostatic pressure, on the arteries when the ventricles are in diastole due to their elasticity. So arterial pressure. Our systolic pressure is the pressure of blood on arteries during ventricular systole. And this is the higher of the two numbers, so you've probably heard your blood pressure, you know, oh, I'm 112 over 78. That diastolic is the pressure of the blood on your arteries during ventricular diastole. And that can kind of give you a measure of the elasticity or health of the arteries. So diastolic pressure of blood on arteries during ventricular diastole. And this is due to the elasticity in our arteries, which is the bounce back, right? Stretch and recoil. Okay. So the systemic pressure on arteries is due to what's going on in the heart. The pressure in capillaries is due to chemistry and physics, and we're going to talk about that in a moment. The pressure on venous blood is coming from the milking action of your skeletal muscles as you move, and from what's called your respiratory pump. So if you recall, we said that veins had valves in their tunica intima, and that as your skeletal muscles contracted, it squeezed on your veins and pushed blood from one valve segment to the next. So that's the pressure that's going on in the venous system, is all this milking kind of action that's happening due to something else. So our skeletal muscles contribute to like the skeletal muscle pump or the muscular pump, and we can say this is the milking action of skeletal muscles pushes blood through veins from one valve segment to the next. So if you imagine this is a vein and it's got these little valves created by the tunica intima, then what can happen is as your skeletal muscles squeeze in, it'll push the blood up through a valve, and then sure, it's pushing back, but when that happens, the valve closes. So we just move one way. And then this muscle down here will squeeze and push the blood up through here and close the valve down there. So that we have the one-way movement of blood from one valved segment to the next. So our respiratory pump is also going to do this milking action on blood vessels, on our veins, but it's happening in the thoracic cage. And as you do inspiration and expiration, there are these volume changes in the thorax that are also going to help to milk your venous blood from one valved segment to the next. So respiratory pump, we could say, is that the pressure changes and volume changes in the thoracic cavity during pulmonary ventilation are going to cause this same kind of milking on the veins in your thorax. So respiratory pump. We'll say this is milking action on veins due to pulmonary ventilation. Gravity also has an effect on venous return. And so it helps with venous return from your head and neck, and it kind of hinders venous return in your lower limbs. So that's why it's really important to be active and not sedentary. Get up and walk around and squeeze those muscles so that your blood moves through your legs. Otherwise, gravity's going to pull it down all day. It's going to pool. You're going to get swelling, and it's going to be no good. So just be aware of that. So gravity pulls muscle or pulls blood down like it pulls everything down. Okay, so what about the astronauts? Let's say they have all kinds of problems to the absence of gravity, but they're still moving their head and moving their lips, and so that will help to milk the blood in astronauts' heads. Okay. Okay, blood pressure. So blood vessels, like everything, cardiovascular system, like everything, is under both neural and hormonal control. So the nervous system is going to be able to do really rapid kind of responses to help with vasomotion, so that we can help to regulate blood pressure. The endocrine system is going to release a whole lot of hormones that are involved when your blood pressure drops, and how it's going to help increase blood pressure is through increasing blood volume. So that takes a little bit longer than our neural controls do. But our neural controls then are going to be using a sensory receptor to tell some sensory neuron about a change in stretch. An increase in stretch indicates that there's been an increase in blood pressure. A decrease in stretch indicates there's been a decrease in blood pressure. That'll go along a sensory neuron to an integration center, who's then going to send efferent messages out to an effector. In this case, the smooth muscle in our blood vessels. And really, the sympathetic nervous system is going to have the greatest impact on blood pressure through its vasoconstriction and vasomotion of our blood vessels, so that we can shunt blood around the system. So let's think about our neural controls. If it's a neural feedback loop, then we've got our receptor, our sensory neuron control center, and our effectors. So what's going on here? If we have a drop in blood pressure, which I've told you is more serious, this is going to cause a drop in blood flow. So this is detected by baroreceptors. They detect level of stretch in your aortic arch and your carotid sinuses. So the sensory receptors are there in your aortic arch and your carotid sinuses, and they are going to detect this decrease in stretch and send the information back to your vasomotor center. And then we will send out efferent commands to constrict our blood vessels. So the vasomotor center then is going to stimulate vasoconstriction. Because if I decrease the diameter of the blood vessel or of a tube exerting a force on a fluid, then the pressure on that fluid increases. So that's going to be a neural control that's going to be really quick. All right, well, what about if we have an increase in blood pressure? So if there's an increase in blood pressure, this is also detected by baroreceptors in your aortic arch and carotid bodies, and they are going to send the information back to your medulla oblongata, the vasomotor center. It's going to send out an efferent command, and now what we can do is if we need to drop our blood pressure, I can vasodilate. So now if I was here exerting this much pressure, if I come out, then there's less pressure. So the vasomotor center here is going to stimulate vasodilation. It's going to be my most direct, fastest way to regulate blood pressure at the blood vessel level. Next, we'll talk about hormones. Okay, so let's think about the hormonal controls of blood pressure. And if you recall, when we talked about the endocrine system, you already met some of these. So antidiuretic hormone can increase blood volume. Aldosterone can increase blood volume. If you increase blood volume, you increase blood pressure. Furthermore, if you decrease blood vessel diameter with that increase in blood volume, you increase blood pressure even more. So when you bring both on board, you can really increase blood pressure in response to a drop in blood pressure to ensure that we have appropriate blood flow. So let's just think about hormonal controls and all the hormones that we've discussed and how are they going to have an effect on our blood vessels. So epinephrine and norepinephrine, which are coming from the adrenal medulla, as hormones in response to sympathetic nervous system activation, are going to be able to stimulate vasoconstriction. They're also going to be increasing our cardiac output by increasing heart rate and contractility, but at the blood vessels, we can stimulate some vasoconstriction. And this is actually really kind of an interesting thing. We said that the veins really aren't good for vasomotion. They don't have big tunica medias. But if you are in such a severe fight or flight situation that you are just flooding the system with epinephrine, then there are some adrenergic receptors on the tunica media and veins that can respond for venous constriction, but you have to be like flooding the system. So, okay, that's epinephrine and norepinephrine are going to do that. What else have we talked about? Angiotensin 2 is going to also be a vasoconstrictor. So that will help by, again, stimulating the constriction of our tunica media. But now angiotensin 2 is also going to be able to stimulate other effectors in the system to help increase blood volume. It's going to go to the zona glomerulosa of our adrenal cortex and stimulate aldosterone release. Aldosterone is going to be another important hormonal control we'll talk about in a second. Angiotensin 2 is also going to go to the hypothalamus and stimulate antidiuretic hormone release. That's going to be another important hormone we'll talk about in a minute. So angiotensin 2 is really, really good for helping to increase blood volume, blood pressure in response to a drop in blood pressure. So it's a great hormonal control for that because it can stimulate vasoconstriction and because it can also help increase blood volume. So the other thing that it's going to be able to do actually is stimulate your hypothalamus and stimulate what we call dipsogenesis, where it makes you get thirsty. So angiotensin 2 will also make you get thirsty. If you get thirsty, then you drink more water and you increase your blood volume yet again. So angiotensin 2, we said, stimulated aldosterone release. What does aldosterone do? Okay, so aldosterone is going to target the kidneys and they're going to reabsorb sodium. And with sodium, water passively follows. So this is going to be another way to increase blood volume. So I get thirsty, I drink more. I vasoconstrict, so now I've got more fluid through smaller vessels. I increase my blood pressure. I stimulate sodium reabsorption, water follows. I increase my blood volume. Antidiuretic hormone, same thing. It's going to target the kidneys and help to increase blood volume, but now we're just going to directly stimulate water reabsorption. So antidiuretic hormone is going to stimulate the kidneys and they'll increase their water reabsorption. And now, through these mechanisms, we can increase our blood volume. So those are the hormones that are going to be able to stimulate effectors in the system when there's a drop in blood pressure, which is by far physiologically more significant than an increase in blood pressure. Because again, if you have a drop in blood pressure, you're going to get a drop in blood flow, and with a drop in blood flow, you have tissue death, body death, death. So, okay, what about if we have an increase in blood pressure? Well, we said if we had an increase in blood pressure, did we say this? We better have. If we had an increase in blood pressure neurally, in the neural control pathway, it was going to stimulate the vasomotor center to allow for vasodilation. Hormonally, if there's an increase in blood pressure, this is going to stimulate the atria of the heart to contract. This is going to stimulate the atria of the heart to expand, and this increased expansion over time due to hypertension is going to cause them to release this hormone called atrial natriuretic peptide, which helps you urinate, take off blood volume, and helps to increase vasodilation, help decrease blood pressure in that way, so by decreasing our resistance. So if we have an increase in blood pressure, this is going to trigger the atria to release ANP, the atrial natriuretic peptide, and it's going to do pretty much everything opposite of the renin-angiotensin system. So it will inhibit our aldosterone release. So we would inhibit aldosterone release. We'll inhibit antidiuretic hormone release. We would stimulate the kidneys to excrete water. So if we excrete water, then we're going to increase our urine volume. And if we increase our urine volume, we're going to decrease our blood volume. If we decrease our blood volume, we help to decrease our blood pressure. And then atrial natriuretic peptide can also stimulate some vasodilation. It's going to, just help in that regard. If we have an increase in blood pressure, the neural controls, again, are going to stimulate vasodilation, and they're going to do that by decreasing our sympathetic nervous system output. Okay. That's the regulation of blood pressure from that mechanism. The other thing, the kidneys can help to regulate blood pressure all on their own, because if there's a drop, well, they're going to sense that and release renin, but they can also decrease filtration of blood. And so if you decrease filtration rate, you decrease urine production rate. If you decrease urine production rate, that also helps to increase blood volume. So the last kind of mechanism that we can use to help regulate blood pressure is, again, in response to like a decrease in blood pressure. What we would see is that we would have a decrease in, it's called glomerular filtration, or glomerular filtration rate. So a decrease in filtration in the kidney, which is going to cause a decrease in urine production. And if you do that, you increase blood volume, which would help to raise blood pressure. So if you had an increase in blood pressure, you could imagine the reverse is true, that we would increase our filtration, increase our urine production, drop our blood volume, drop our blood pressure. All right, the last thing that we need to talk about is capillary exchange. Capillary exchange is the movement of material to and from the blood and tissues. So it's the two-way movement of material, because we're moving out of the blood to the tissues, and then from the tissues back to the blood. Blood is flowing through capillary beds in one direction, from the arterial side to the venous side, but there's movement of fluid in that capillary bed. So there are going to be three important processes that are involved in capillary exchange. Diffusion, one that's called transcytosis, and then one that's called bulk flow. And so let's talk about each of these and what pressures are guiding the movement of fluid into and out of the blood and tissues. So transcytosis, trans means across, cyto means cell, osis means movement. So transcytosis is when you bring something in through a little like pinocytotic vesicle and bring it into the cell that way. And so this is, you know, we said that the inner layers of the blood vessel wall were nourished by the blood flowing through them. So that's one way you can get stuff into you, is bring it in. And so then it's into you, and then we can, you know, do whatever we need to do, produce whatever we need to do. We can let it out on the other side. So then we're across and out. We would let it out by exocytosis. So we're really not going to say a whole lot about transcytosis other than that. So this is movement of material across the cell. So by vesicular transport. So we could say this is movement by vesicular transport. Diffusion. What's diffusion? It's a passive process where we move from high concentration to low concentration. And we can either have simple diffusion where you go right across the barrier because the barrier is permeable to you, or we can have facilitated diffusion where you stick a protein up in the membrane and facilitate the diffusion of something. Either way, it's the movement from an area of high concentration to low concentration. So diffusion. And in diffusion, this is actually the most important method of capillary exchange for the tissues. It's going to be the second least amount of time that we spend talking about a process, though, because really the most amount we're going to spend on the next, because there's two kind of parts to it and it takes a long time to wrap your head around. But this is most important. Why? What do tissues need? Glucose and oxygen. Glucose and oxygen both use diffusion. They're small enough that they can squeeze right through here. Cells use oxygen and glucose, so it's very easy to take them up into cells, and it's very important for our tissues out here. So diffusion is the most important for the tissues. It's a movement from high concentration to low concentration. And it's going to be how our glucose enters the interstitial fluid and then can be taken up by cells, and how our oxygen enters the interstitial fluid and then can be taken up by cells. So as far as the tissues are concerned, this is kind of the most important thing that's going on. So this is what we'll say our small solutes like glucose and oxygen can use this. All lipid soluble substances can also use diffusion. All right. Then, what else? That's really all I want to say. CO2 also diffuses, but the tissues don't need CO2. They need it out. So it diffuses back into the blood, and then we can take it off to the lungs, where we can easily diffuse across the alveolus. We'll talk about that when we get to the respiratory system. So the last mechanism of capillary exchange is bulk flow, baby. Bulk flow, baby! Okay. So, the last mechanism of capillary exchange is called bulk flow, and it gets that name because this is how we're moving large quantities of fluid and all of the dissolved solutes from the blood and tissues. So there are kind of two little smaller parts to bulk flow. So bulk flow has what's going on the arterial side and what's going on the venous side of a capillary bed. So here in orange, this is our capillary bed, and on the arterial side, we're bringing in this blood that is oxygenated. So it's got a high partial pressure of oxygen, low partial pressure of CO2, it's high in nutrients. And if we were to look out here in the tissues then, the tissue cells have a high partial pressure of CO2, a low partial pressure of oxygen. They're low in nutrients, high in wastes. And so now we need to have the movement of these fluids into the tissues and then out of the tissues back into the blood. So what's happening here in bulk flow? On the arterial side, we're using hydrostatic pressure to push against this semi-permeable membrane. What passive process is that? It's filtration. So filtration is the process that dominates bulk flow on the arterial end of a capillary bed. So filtration. And what we could say is that this is due to the hydrostatic pressure out, which is coming from ventricular systole. So when you get here, you still have some pressure from the heart contracting. And when you get into the capillaries, we're going to disperse it, but that's what's going to push all of this fluid out on the arterial side. So we could say this is filtration due to hydrostatic pressure out from ventricular systole. And this is going to be how we get all of our fluid out there. So in here it was blood plasma that was high in nutrients, high in hormones, high in drugs, high in alcohol, high in oxygen. And so we're going to push against this semi-permeable membrane and push all of that stuff out here. And then now our cells are going to take up what they need. And on the venous side now, we have a different pressure. When we go through this capillary bed, we disperse all of that hydrostatic pressure out due to the ventricles contracting. So what's going to drive the pressure back in? Well, let's think about this. What does this blood look like after filtration? Is it hypertonic or hypotonic? It seems hypertonic because it's full of all these large blood cells that didn't get pushed out and all of these large proteins that didn't get pushed out. So now the pressure on this side is not going to be hydrostatic pressure, it's going to be osmotic pressure. We have a pressure on water to move from the interstitial fluid back into the capillary bed. So that's what we see on this side. We call it reabsorption. And that's going to be the process of bulk flow that dominates on the venous side of a capillary bed. So over here, we could say we have reabsorption from the tissues to the blood. Reabsorption is going to dominate movement on the venous side of the capillary bed. And we'll say this is due to osmotic pressure in because the blood is hypertonic. And water always wants to go from hypotonic to hypertonic. So you have osmotic pressure in the whole way across the capillary bed. It's just true. But out here you have a lot of hydrostatic pressure out, so we get a net movement of materials out. And then the osmotic pressure is going to drive about 85% of our fluid and dissolved solutes back in. This is where those lymphatic capillaries came into play. The lymphatic capillaries that we talked about at the beginning of last lecture are sticking in here and picking up all of our extra interstitial fluid and returning it to the cardiovascular system through the subclavian veins. But we'll talk more about that when we get to the lymphatic system. So if you look, there's a figure in your book that will show you all of the pressures. You'll see your net filtration pressure out, and then we'll see our osmotic pressure back in. I'm not going to have you calculate those, but I do want you to understand the movement of materials on each side of the bed. So if I were to say something to you like, most nutrients move to the tissues using this, then the answer would be A, diffusion, because glucose is what our tissues are using. But if you're thinking of nutrients as things like ions and vitamins, then we could also use filtration from the blood to the tissue. If I were to ask you, how are we removing waste from the tissue? What process? then you would say, oh, well, I'm going to argue that CO2 is a waste gas, and that's going to use diffusion. Okay, fine. But what about all the rest of our waste, like urea and everything that gets left behind from cellular respiration? How does that get picked back up? Reabsorption from the tissue to the blood. And that's what dominates on our venous end. Filtration dominates on the arterial end. Be clear on that, because I guarantee we'll be tested on that. And that, my friends, wraps up the cardiovascular system, except for that I had a question for you I just skipped over. So here's one last question for you. Most material returns to the blood using A, diffusion, B, filtration, C, reabsorption, D, lymphatic capillaries. Oh, you're right, reabsorption. Now what if you say, yeah, but you just told me lymphatic capillaries return the extra interstitial fluid to the blood? I would say you're right, 15%. That 15% is huge if it doesn't get picked up. Imagine you have 15% of that blood gets left behind in your arm all day because you had, I don't know, breast cancer and they removed all your lymph nodes and lymphatics. Then that 15% is huge and you swell and it's life-changing. So they have to give you compression sleeves and stuff like that. So yeah, it's a lot of fluid, but it's not the most fluid. 85% is more and 85% is reabsorbed on the venous side of a capillary bed. All right, it's been fun. I'll see you next time. Dissolved substances to some bulk flow for you.